is my pleasure to now introduce our first speaker in our segment focusing on cutting-edge neuroscience research, Dr. Kay Tai. Dr. Tai is a professor and Wiley Vail Chair of the Systems Neuroscience Laboratory at the Salk Institute for Biological Sciences and an adjunct faculty member at the University of California, San Diego, UCSD. Her research program is focused on understanding the neurobiological mechanisms underlying social and emotional processes at the level of synapses, cells, and circuits, and particularly those relevant to psychiatric disease. A very timely topic. Kay earned her PhD in neuroscience at the University of California, San Francisco, UCSF, in 2008, and trained as a postdoctoral fellow at Stanford University with Dr. Carl Diceroth from 2009 to 2011. She then became an assistant professor at MIT in 2012 and was promoted to the rank of associate professor with tenure in 2018. She recently moved her laboratory to the Salk Institute in 2019. Kay has been recognized with many prestigious research awards, including the NIH Director's New Innovator Award, the Presidential Early Career Award for Scientists and Engineers, PCAS, the Society for Neuroscience Young Investigator Award, Technology Review's Top 35 Innovators Under 35, and the NIH Director's Pioneers Award. She has also been recognized with a number of awards for mentoring at the undergraduate, graduate, and postdoctoral level. Kay is committed to outreach, promoting diversity and inclusion in science. Kay, on a personal note, I still remember the first time I met you in New Orleans in 2012 during the Society for Neuroscience SFN annual meeting. You had, I think, just joined MIT and were starting up your lab. We had just founded Inscopix literally a year earlier, and we're launching our first product in Vista at that SFN when we met. I remember being struck by how visionary and passionate you were. You wasted little time in connecting the dots between what we were demoing that year and the research questions you wish to pursue. It's truly been fascinating watching you from afar, growing in your career as an accomplished professor scientist, and also becoming a role model for young women scientists around the world. Kay, it's a real honor to have you with us today to speak of the neurobiology of social isolation, something that I think we're all struggling with in this COVID era. Kay, the virtual stage is yours.
Thank you so much, Kunal. It's such an honor to be here today, and with this incredible lineup and the mission that you guys are focusing on today is something I feel really passionate about. So I'm just going to pull my slides up right now. All right. So again, it's a huge pleasure to be speaking today, and actually, I'm going to unveil some very, very new data that literally we've just been, working over the weekend to finalize the plots and replot them so that we can show them to you today. But first, I want to start with a call to action and something that's very important. So I just want to start by opening with a public service announcement about why Black Lives Matter is really the only logical message. And this is really due to the minimal correction to account for the massive yet immeasurable accumulation of disadvantages that Black people in America will experience over their lifetime. So here I'm showing you data that unarmed Black people were killed by police at a rate of 5 times that of unarmed whites. This is not correcting for the difference in population. So this is much more severe than it even appears here. So from this, we already seem to know that white lives matter. And so therefore, Black Lives Matter is the only new non-redundant information that we need to communicate and internalize. So we would never apply a linear filter to a non-linear data set. And why would we do that here? So that is my call to action. This is the July 4th, 2019. This is July 4th, 2020. 2019, 2020. Social isolation and even just perceived loneliness correlates with deficits in mental and physical health, a shorter lifespan, specifically increased anxiety, depression, suicidality, and a number of additional other mental health disorders. And also physical health consequences such as increased morbidity and mortality for diseases like cancer and heart disease. increased inflammation, and particularly noteworthy, increased symptom severity with a viral immune challenge. So now more than ever, there's just an urgent need to understand the neural circuit changes underlying perceived loneliness or social isolation to prevent these negative effects. So this comes back to a bigger question that I've been interested in for a number of years now. And that is, many of the most successful species on the planet engage in complex social hierarchies with social structure and stability that offer safety, homeostatic needs, satisfying, opportunities for play, for mating, for warmth. And one of the things that has fascinated me the most is, even in the face of changing environmental conditions and changing internal composition, somehow individuals know their place in their social rank. They know their place in the social hierarchy. And how does this happen? So this is a concept, many of you are probably already familiar with the concept of homeostasis, which just comes from the Greek words for same and steady. And this just refers to any process that organisms use to actively maintain stable conditions necessary for survival. In its simplest form, there are a couple of different ingredients that we need in a homeostatic system. And I want to say that there's been beautiful work done in neuroscience looking at energy balance, thermoregulation, osmoregulation, and a number of other types of homeostatic systems. Each of these homeostatic systems has a detector, a receptor that detects the change. For example, if we're thinking about thermoregulation, something that detects my body temperature. If my body temperature is above the set point that is held within the control center, and the detector feeds this information about my body temperature to the control center, if my control center then computes that there's a surplus or a deficit, for example, say I'm too hot and I'm over 98.6 degrees, which is my control center set point, then I will activate an effector system, which might be sweating or moving into the shade. That is essentially my response that interacts with the environment to correct the change. Then my body temperature comes back to normal and it shuts the whole system off. So this is the system for thermoregulation, but what about social homeostasis? And so I really want to highlight the work of Gillian Matthews. Gillian Matthews is a brilliant visionary who is a former postdoc in my lab. She just left my lab last week to start her own lab in the UK. So if you're interested in this work and interested in a postdoc in the UK or any sort of scientific position there, I encourage you to reach out to her. And what Gillian found a few years back was completely serendipitously that there are these dopamine neurons in the dorsal raphe nucleus. And if you isolate a mouse for 24 hours and then record from these dorsal raphe nucleus dopamine neurons, that you see an increase in excitatory input or glutamatergic synaptic strength onto these neurons. Additionally, after 24 hours of isolation, you will also see an increase in the amount of neural activity that you see from GCAM. In terms of this genetically encodable calcium indicator, which I'm sure you'll hear all about. And so in response to a social stimulus, you'll see a greater response after isolation than animals that are group housed or than animals that are isolated but exposed to a novel object. And so all of this set of data just goes to show that dorsal raphe nucleus dopamine neurons are sensitive to acute social isolation and track social isolation. And this is just the graphical abstract from Gillian's 2016 Cell paper. And the next thing we wanted to do was optically manipulate these neurons using optogenetic tools. And when we photostimulate these neurons, we found that, yes, there was this increase in sociability, an increase in motivation to seek social contact, essentially a pro-social effect, but that photostimulation was actually aversive and made animals want to avoid a place paired with photostimulation. Initially, this was a little bit confusing because, ventral tegmental area dopamine neurons also, when stimulated, become pro-social, but are highly reinforcing, and animals will work very hard to receive photostimulation for VTA dopamine neurons. So this result, which we replicated a number of times, really baffled us at the beginning. Fortunately, we could draw on the conceptual framework from our feeding work, where you could have multiple valences involved to engage in eating. You can eat this delicious dessert, even though you don't really need it, because it's rewarding, because of food reward, and you enjoy it. You can also eat something that's maybe not so palatable just to escape the unpleasant state of being hungry. Similarly, you can engage in social contact because it's rewarding, or you can engage in social contact to escape the unpleasant state of loneliness. And loneliness is a universally unpleasant state. Solitary confinement in humans has been deemed torture. And so this is something that we started to think about. Furthermore, following acute isolation, many animals, humans, non-human primates, rodents, will show an increase in social interaction upon reintroduction to the social group, essentially an isolation-induced rebound. And so this isolation-induced rebound was blocked by photoinhibition of these dorsal raphe nucleus dopamine neurons. So this, the final component of this is that if you look into the psychological definition of loneliness, and this is a term from psychology, there are a number of different criteria. And the trickiest one for us to meet in animals was that loneliness is subjective. And what does that mean? That just means that you can be lonely on a crowded street or in a crowded place in Manhattan, but you can also feel not lonely when you're alone. And so that is very much the subjectivity. So how do we get at this in mice? It's extremely challenging to do so, but we wanted to be able to look at something that, sort of discounts the fact that social stimuli are so much more interesting potentially than other types of stimuli. And so one thing that we looked at to get at the subjectivity is to look at social rank. And so what we found is that social rank strongly predicted the behavioral effect induced by both the photoactivation manipulations and the photoinhibition manipulations. And so this was something really interesting. If you allow me to wildly speculate, I'll just say that we were thinking perhaps a dominant animal really enjoys their social environment. They have prime access to resources. All other animals are engaging in affiliative behaviors. So when a dominant animal is isolated, perhaps they do feel, quote unquote, loneliness or a loneliness-like state. However, a subordinate is being denied prime access to resources, getting beat up all the time. Perhaps when this animal is isolated, they don't feel lonely. They feel relieved to be away from, their bullies. And so this can be a way in which we can get at the subjective aspect of loneliness. And of course, that is, again, speculation. Altogether, what I think we've seen from our initial study is that dorsal raphe nucleus dopamine neurons may represent an effector system for social homeostasis. We're also very excited that our effect was recently replicated in humans. Just this general increase. Of course, it's not at a cellular resolution. This is bold fMRI, but this general phenomenon is also found to be true in humans with just 10 hours of social isolation. And this was done with our collaborators at MIT, Olivia Tomova, in Rebecca Sachs's lab. And this was pre-printed already, so you can find it online and by our archive. And it'll be coming out soon in Nature Neuroscience. So now on to some very exciting preliminary data. What we've found now is that dorsal raphe nucleus dopamine terminals project to several different downstream targets. The projection to the central amygdala when we do this sociability assay shows that when we photostimulate dopamine terminals in the central amygdala coming from the dorsal raphe nucleus, that we increase prosocial behavior. And importantly, even in this specific input, we also find that it is indeed correlated with social rank. Really, the effect that we see are driven by the dominance. We don't see this effect in projections to the BNST, nor projections to the posterior portion of the basolateral amygdala. So next, to probe the impact of dorsal raphe nucleus dopamine terminals in the central amygdala, we used Invoke. We've used Invoke before, and Caitlin Vanderweel in my lab, along with the rest of our team, has validated Invoke with in vivo electrophysiology in her 2018 Nature paper. Now, Christopher Lee in my laboratory, a graduate student, has worked hard with Jillian Matthews to do this following experiments and look at what happens when we stimulate these inputs into the central amygdala. And so the task is pretty simple. It's basically a 3-chamber where there's a mouse on one side, and not on the other. There's a little grill divider here. And whenever animals enter this social zone where they're investigating the other animal, or this non-social, we'll count that as an event. And 50% of the time when they enter the social zone on some of the trials, we will photostimulate deer and dopamine input to the central amygdala. And we're looking here at what central amygdala neurons do with and without that stimulation. And what you can see here, we just cluster the neurons that we have. So we're looking at movement initiation, the juvenile zone or the other zone, and contact with the divider. And so what I want to draw your attention to is right here. So in cluster 1, we're doing a hierarchical agglomerate functional clustering. And what you can see is there's one cluster of neurons that becomes active when the animal is engaging or approaching the juvenile, the social zone. And this is specific to when there's no photostimulation. These neurons are not active when we do photostimulate. And then look at cluster 2. Cluster 2 is the other component here, not active when the light is off, when the animal's interacting with the juvenile, but are active when the light is on. So what could this mean? How do I interpret these data? The way that I think about this is that dorsal raphe nucleus dopamine input is mimicking a loneliness-like state from our previous cell paper. And then what we can see here is that when an animal is in a loneliness-like state or not, it is using completely distinct, different ensembles to represent this social stimulus. So, wild speculation would be thinking that the way that my brain, I'm using different neurons in the central amygdala to recognize or to engage with social stimuli when I'm lonely versus when I'm not. Now, thinking about how this pertains to the big picture, and this is my last slide, when you experience acute social isolation, you detect this deficiency with your control center. Then you ultimately activate your effector system and then rectify that deficit until it's back at your homeostatic set point. But in a case like what we're in today, where we can engage in social isolation, and there's no way to really correct for this, this deficit, what can happen with chronic isolation is a set point adaptation. And this set point adaptation switches animals from an acute isolated state, wherein animals are more social, pro-social, when they are reintroduced to the social group. After chronic isolation, animals become antisocial, more aggressive, more territorial, avoidant when they are reintroduced to their social group. And we hypothesize that this is due to an adaptation in the social homeostatic set point. And so this is something to pay attention to soon. Christopher Lee and I are composing a review that will be out in Cell soon. And with that, I'll just close and thank everyone in my lab who's contributed to this work. Past and present are many wonderful collaborators and funding sources, and you for your attention. Thank you.
Thank you so much, Kay. That was a fascinating talk. We have a few questions from the audience, but just to get started, picking up on your last point, if we could, speculate, what would you say are some of the implications as, we as a society, hopefully in the near future, start emerging from social isolation and loneliness and, quote unquote, reintegrate ourselves with society? I mean, do you, for example, fear that, as a society, we will become more aggressive because of the loneliness we have been in over the last, few months as we reintegrate ourselves into the broader society post-COVID?
I mean, I think that's something to be aware of. This aggression phenotype is something that you've seen in humans, non-human primates, even Drosophila. So I think this is something that's very well conserved across evolution. I think in many cases, the reintroduction that we're experiencing right now, phase 1, phase 2, is sort of doing that. And so in a way that might already be implemented for other reasons, but it's just something to be aware of. And our working model is that when we have this homeostatic set point adaptation, this is possibly what could be triggering this, cascade of mental and physical health consequences. So that is our current working model, but so much remains to be known. It's a very embryonic field of research.
So interesting, so interesting. So a question here from the audience, does the synaptic rewiring associated with homeostatic stability have side effects such as on memory correlation?
That's a great question. We have not tested that. We currently don't, as of yet, have any reason to think that DR and dopamine neurons are necessarily directly important for certain types of memory, but that has just simply not been explored. Dorsal raphe nucleus dopamine neurons are a relatively new cellular substrate for investigation. Mm-hmm. There's only a handful of papers about them. So that is an open question.
Got it. Another question from the audience related to the coping mechanisms or the need to socialize amidst lockdown. Is there a learning rule at the microscopic level or the plasticity that could explain this?
A learning rule for the homeostatic change? I
think that's what the question might be pertaining to, yeah.
Oh, that's a really, really fascinating question. It's interesting. I've kind of been framing it in terms of homeostatic systems, mm-hmm. not necessarily like conscious learning. But that's a really great point, and I hadn't thought about that before. But I suppose this homeostatic conceptual working model could also be reframed in terms of thinking about it as a learning rule. So I really like that, and I think also a very important thing to consider is that if isolation is predictable and expected, that can also temper the negative potential consequences. And I think Byung-Kook Lim at UCSD has done some really great work on this as well.
Got it. A couple of more questions. And I know we're running out of time here, so maybe I'll do one more question and we'll call it a wrap up. This is more of a question specific for you and how you're protecting your lab, which is, I think, a fair question that a lot of PIs have as they try to adapt to these phased mechanisms of post-COVID life. The question here is, what steps are you taking to protect your own health and your lab members' mental health as this pandemic rages on?
That's a great question. It's always such a challenge because we want to balance the immediate urgent need to protect people from, COVID, but also this sort of other, problem in the background. So it's all about finding that balance. And so I think it's been important to stay connected through virtual means, which we have been relying on heavily in 2020, but also to have a bubble and really rely on your bubble. And so I think those are the things that we're focusing on right now, but that I don't have all the answers. It is a constant daily learning experience for me as well.
Thank you, Kay. I think that's a takeaway we can all benefit from, not just those of us who are running labs. Thank you so much, Kay. Really, really appreciate the talk, enlightening, and so honored to have you here today with us. Thank you.
Thank you so much. Looking forward to the rest of these amazing talks. Hi, Carrie. Okay, bye.